s'appelait Amélie.
So now let me call Amélie Piton, and I thank Amélie for coming with us. So here we explore neuropsychiatry, but we are now with Amélie coming back to psychiatry and to neurodevelopment and ASD. Amélie is a genetics specialist. We're going to talk about molecules and physiology, cell physiology. So you spoke about the genetics of neurodevelopmental disorders and especially ASD. And you have a special focus on interaction between the hormone, the androgen, and the genetics of our patients. So please take over. You have 45 minutes. Hello all. Do you hear me? So my voice is a little bit shaky, and I hope I can hold for 45 minutes. So I'm not going to speak too loud, but I will keep like this if it's okay for everybody. So I'm so pleased to be here with this incredible weather. I hope it pops a bit later. I think 6 or 8 years ago, I was already here at the ARAPI to talk about the sex bias in the neurodevelopmental disorders, the genetics, hormone hypothesis, and have we moved forward? Well, you will see that no, not fully, but I will give elements that were found in the past few years in relation to our research projects and also published by other teams. Thank you very much. And we find a sex bias that is more boys impacted. I will focus more on the two that I work on, which are the ASD spectrum and also the neurodevelopmental disorders, which is the new appropriate name for intellectual deficiency. So it's quite important for ASD, 1 girl for 4 boys, less for intellectual deficiency, or 1 for 1.4 boys. And you will find that the success of boys impacted will be found further. So the first hypothesis for this bias is that we do not diagnose the same way. The recruitment is different. So I will not really venture into this field because I know that a lot of more people understand this better. But I will present the biological hypothesis that could explain this bias. So the first thing is genetic differences, that we don't have the same number of the chromosomes. We have the same number of chromosomes, but there is a difference with the sex chromosome. Girls have 2 X, 1 inactivated, and the boy with just 1 X chromosome, and on top of that has a Y chromosome. Does the X chromosome, I will focus on the X chromosome, and can it explain the sex bias in the developmental disorders? And the second hypothesis in the hormone difference. There is an English team that has been working on this for several years now, the influence and effect of testosterone. And why testosterone? Because we know that the peak of production of testosterone is adolescence in boys, and early production peaks in uterine life and at a later stage when the brain is in development. So I will explain first the genetic aspect. Do the genetic data that we have on neurodevelopmental disorders, NDD, this graph represents the number of genes that have been identified so far that are involved in the monogenic NDD, with rare diseases with an expression of NDD. 17,023 genes, 1,723. CISID was the name, intellectual disability, and so now it's called CISNDD. So now we're going to talk about cases of one mutation in one of these genes will be responsible for the mutation and what we call the monogenic form of NDD. So with technological revolution of the high-speed sequencing, we have genes that are involved in the recessive autodominant, recessive and autodominant. So there is a lot of overlap. What Boris Chomet showed you yesterday, there is a big overlap in the ASD and intellectual deficiency disorders. So we'll find patients with a mutation in these genes that present with an intellectual deficiency disorder. So the genetic architecture of this ASD and intellectual deficiency. So you can sequence the genome of one individual. So anomaly of the number of copies that will carry a bit of the chromosome. So we are looking for cases where the genetic anomaly is responsible for the phenotype. So we understand that there is a single genetic anomaly responsible for the phenotype. For ASD, and in less than 10% of the cases, we showed you yesterday that we have clearly, even though the same genes are implied in the monogenic form, we have a difference of architecture between ASD and IDD. So you have this pie chart of the genetic architecture. One anomaly is responsible for, we have identified over 1,700. We have the anomaly of the number of copies, the replication, several genes. So the genetic event, the genetic anomaly responsible for the phenotype. So when we do sequencing, we find this anomaly and we find that it is responsible for the phenotype. And we say in our lingo that it is pathogenic. And you find other things, a genetic anomaly, but it is responsible for the phenotype, but with what we call a PF, that is incomplete penetrance. Some individuals will have this genetic anomaly, which will be responsible for their NDD. And the same with people who present no NDD. We have found this, and we think it explains part of the phenotype, but we need to be cautious because the predictability of this information or the predictive value is really something that we need to question because we cannot predict the clinical manifestation from one individual to another who present the same and then the other side of the pie chart in blue has all the complex forms, but where there is a contribution of the risk factors, genetic risk factor and environmental risk factor, that is. So if you want to look at what is happening between the IDD, the participation of these monogenic forms is very important, and also anomalies of the number of copies that are responsible for the IDD and the complex forms. You may have a small proportion, but which will be not too important. When you do genome sequencing, we find a variation of responsible for the phenotype. Some forms have not been identified yet, and this is the reason why we're just at 50% or not 80%, but we think that majority of the forms will be due to one anomaly. When we do this genome analysis, in less than 10% of the cases, we find this, we think that it contributes to only a small proportion, and we think that there are more complex forms than in IDD. So when we do diagnosis, either we have all the means necessary and we can do the sequencing ad libitum, or maybe in this case, Some genetic factors are involved, but we do not have the means to clearly identify them at this stage, and at least we are not able to give an individual diagnosis and say this is the cause of your ASD. We can only draw genetic information from these cases, and we are trying to investigate why do we have more chances than in complex forms. And in October 2023, so just last year, there was a recent study that looked at the SPARK cohort, that is the American cohort on ASD, and they looked at all the people in where there were pathogenic mutations, and they looked at the associated signs, and they looked at the chances of having a pathogenic cause. So I'm not going to quote all of them, but you have intellectual development disorder. When it is present with an ASD, it increases the chances of finding a pathogenic mutation. A motor delay, a microcephaly, increases the chance of finding something more than macrocephaly. Small size, small stature, or eye squinting, or face traits that will increase the chance of finding. So they looked at the rate of diagnosis, that is, in how many cases do we find a pathogenic mutation. If there is no zero sign, one of these signs, 2 or 3 or more. And you see a drastic effect on the, when you have none of these signs associated with ASD, you find they find in their cohort 4% of diagnosis. And when you have one sign, it jumps to 8%, and 2 signs, 3 signs and more, 21%. So trying to find consensus with my colleague Boris Chomet, I would propose that please do sequencing if you have the possibility to, but if you have them, do not hesitate to send the dossier to us, that is with 3 or more, maybe these, maybe those, because we will have a larger chance of finding in case 4. Do not hesitate genetic sequencing or genome sequencing for the cases where you have 3 or more. And it's important to have that genetic diagnosis. Out of these 1,700 genes, we have dominant autosomic forms, a mutation on one of the chromosomes, on one of the copies of the gene that causes the pathology. So I showed you here one of the genes on which we work on. It's a nice illustration of this overlap between IDD and ASD, a syndrome DYRK1A. So this gene DYRK1, it's going to regulate a lot of cells in the protein. As Chomet evoked yesterday, these mutations are de novo. That is, they're not present in the copies of the genes of the parents, but they will take place here in the copy of the genes in the child. The clinical manifestation, you have language disorders, smaller head, and kind of a physical resemblance in the faces. The traits of the face that are peculiar, and this should orient toward genetic seizures or epilepsy or ASD half of the time, and this was something we're able to demonstrate, and we use 2 methods to see the percentage of ASD. We have a questionnaire, a database where parents can enter the name of the gene affecting the child, and they can answer clinical questionnaires. And in half the cases, in half the responses that we had, the answer is yes. And we also have Benjamin Durand, an intern in Strasbourg, who had conducted interviews with parents, conducted several tests, and in 57% of the cases, a diagnosis of ASD was made. When you look at this form in particular, of rare diseases of the neurodevelopment that associates ASD and IDD. We have no difference between boys and girls, just as many boys as girls. And so at this stage, we don't have a self-evident or very obvious difference of severity of the clinical manifestation between boys and girls. And this is important for the rest when we are going to talk more about the bias, sex bias. So the mode of transmission of these genes are the mutations on the X chromosome. The hypothesis is that are these mutations on chromosome X, could that be responsible for the excess of boys? So this is a more or less typical family with an NDD related to the X chromosome. So you see that it is transmitted by the mothers who are asymptomatic, even if in some cases they can be weakly affected, maybe not a full-blown NDD, but some difficulty learning or nothing at all. So they have two X chromosomes, one with and one without the mutation, whereas the boys have only one X chromosome, and if it bears the mutation, the girl will inactivate the X that carries the chromosome, the mutation. It's not always the case. So it's a very basic vision of this picture. And we know that we have this form, and in this family you have 3 boys. Can this explain the sex bias that we have in the... So just to illustrate these families where you have this kind of, the neurolichen 3, neurolichen, this was published by Thomas Bourgeron in 2003. It was one of the first ASD-implicated genes that we could call non-syndromic. 2 boys with ASD with variable severity. And they identified a variation that changed an amino acid in position 451. And it was published, and then nothing. And a lot of people sequenced this gene, but did not necessarily find a mutation. But we sort of randomly found another variation that doesn't affect the same amino acid, but which is also in the neurolichen 3. In one boy and in 2 of their maternal cousins to the transmission by the mother. So this protein types, it codes a synaptic protein, the neurolichen 3, that enables the contact between neuron and the synapse at the level. So what we were able to show is that the protein without mutation will be located on the surface of the cell. But when there is the mutation that is identified, we don't have it on the surface of the cells, and the protein is maintained in some compartments of the cell and does not reach the membrane. And we found other variants with ASA, ASD, and IDD, but we're compiling everything that we found in the 21 years. We have 10 individuals maximum, which is not enormous. So that does not explain for the sex bias in IDD, in neurodevelopmental disorders. So if you look at all the genes that could lead to a neurodevelopmental disorders, you have more than 130 genes on chromosome X. So genetics tells us, when you look at the genetic analysis with in persons with TND, it says a different story. It says that we find mutations on in 6 to 8% of boys, but also 6 to 8% of girls, which means that we don't necessarily have a variation that is pathogenic in boys more than girls. And why? Well, actually, we have this form neurolichen 3, which affects mostly boys, or girls would be if there's an accident on the other X chromosome. And we also see forms that affect boys, but also girls. Fragile X. Girls can be affected if they have an impact on FMR1 gene. and forms that affect mainly, mostly girls. So on this DDX3X, a gene identified a few years ago, the girls will present a de novo mutation, and the most frequently mutated gene in girls is on the X chromosome. So there's no explanation really for the sex bias. So this was established that there is a similar contribution in boys and girls of the monogenic forms that are identified today in relation to the fragile X at this stage. And some studies tried to go further to see if there were other factors that have not been identified. So this study I conducted a few years ago in Canada. We decided to sequence candidate genes on X chromosome in a cohort of persons with ASD. And conducting the analysis, we thought we would find something in boys, but we only found 2 mutations that we considered pathogenic, and we found them in 2 girls. So the notion was that we did not have an explanation for the sex bias. And we have 2 colleagues in Paris who took families, 12 families with boys with ASD, and high frequency sequencing enabled her to do this, but the result was disappointing because she couldn't find a potential risk factor. So even if we put more boys in our cohort of boys with ASD or NDD, and the first conclusion is that mutations on the X chromosome in the monogenic forms does not explain the excess of boys. The monogenic forms contribute more to NDDs in girls than in boys. If you look at boys with a IDD, so these are the results that we obtained in Strasbourg, where we sequenced a panel of genes, either the whole genome or the coding genes or the genes implied in the NDD. I'm very, blue for boys and pink for girls. So you see that there is an excess of boys. This was not something that we sought to deliberately establish. When we found a mutation of a gene responsible for the NDD, it's always more in girls than in boys, and with about a 10% difference, either as a panel, exome, or we find more mutations in girls than in boys. So that was for IDDs, but for ASD, in that a paper from last October, what I showed you yesterday, a few minutes ago, we had in dark blue the girls and in green boys. And you will see that you have always more diagnoses identified in girls than in boys. So we have this discrepancy of 10% between boys and girls. So if we look at all of this, what does it mean in genetic terms? It means that in 10% of monogenic forms, we find them more in girls than in boys in genetic events that explain the NDD in boys than in girls. Explained the excessive anomalies of the number of copies in girls with ASD. So we could imagine the number of the boys impacted is not due to a monogenic reason. So the hypothesis would be that for the DRK1 gene, a form that is monogenic, same number in boys and girls, but the contribution is not the same when you relate this to the total number of boys affected, the total number of girls affected. In these boys, there is an excess of more complex forms that would account for this sex bias. So if we try to understand why there is an excess number of boys, so maybe it would be interesting to go look in these monogenic and maybe investigate more in the complex forms, whether we have environmental or genetic factors, and the environmental factors could be related to hormones. And the second part is the question of, is an early exposure of boys to androgens, could that be responsible for the sex bias in the NDD presentations? So this is the work that has been conducted by this team led by Simon Baron-Cohen in England. And he's been working on this for several years and tried to show the association between autistic traits and the rate of prenatal testosterone. He shows the value of the QChat. I will not justify the choice of that score, but the rate of prenatal testosterone and sees a positive correlation. And they also showed that there were more autistic traits in the descendants of women with a level of testosterone that is high. The polycystic syndrome, ovarian syndrome. And there is also a differential expression of the enzymes implied in the metabolism of steroids in the persons with an ASD. So there is an early production of androgens, and it's implied in the development of the brain. You see the development of the brain here with the various phases that are important for development, the proliferation of neurogenitors. So we could imagine that testosterone does play a role in one of these important stages. So we tried to look at that. So here is a diagram on the action of testosterone, or it's a dehydrogen testosterone, which is a powerful agonism to the work of the receptors. Testosterone can be transformed into androgen. So this is, we call it DHT, and it's the one that is used more, DHT, that is links to the receptor, and then we'll relocate in the nucleus of the cell, and we'll regulate the expression of the genes, that is the replication of the messenger RNA. So we tried to look if the androgen could disturb the processes or regulate the genes that are involved in neurodevelopment. And we have a model used in our lab, an in vitro neurons that can multiply in a Petri dish. So we used these human progenitors and wondering whether it could affect the proliferation of these progenitors. And the last question, will this affect the regulation of genes implied in the development of the brain, or even genes that are involved in ASD or IDD? So it's going to relocate in the nucleus. In blue, it's the nucleus, and green is the receptor. You see that all cells express very strongly the receptor to androgen. And when you put DHT, you find that they will overlap, and it means that they relocate in the nucleus. You have to understand that the progenitor is more strongly expressed, and what we wanted to see is whether this treatment of DHT will promote the capacity of multiplication. So this is what we are able to demonstrate. This curve here, the growth of the progenitors and their multiplication in the absence of treatment, and in the dotted line, you see a strong increase of the proliferation capacity of the cells. And then we wanted to see if this effect was mediated by the receptor to androgen. And that enabled us to degrade. That is, we had no longer had fabrication of this protein, that is reception. And the red line shows that we have no longer an effect of the DHT treatment, which means that the mediation by the DHT is really strongly established. And we also wanted to look at the effect of androgens on the proliferation capacity of these neuronal progenitors in a Petri dish. And so we induced a differentiation of these cortical neurons. You see in the green box the neural networks being shaped. In normal conditions, we don't really have an effect. In normal condition, we see the same proliferation. But under stressors, where we remove from the cells, we remove the cells from a milieu where they can differentiate and survive, we no longer have neurons. All these cells die and no neuron formation occurs. But if we have a DHT supplementation, we have a survival of the neurons, and we can recover some neurons that will differentiate, not as much as in normal conditions, but still a bit of differentiation. So under stressor. And we also wanted to see if genes were mediated by DHT and what it does on the expression of genes. And you see that the result of this gene expression, genes that are underexpressed and overexpressed, and this is the value, the p-value of the difference that we have between the two conditions. And we have certain genes where we have a very strong increase. So the gene NRCAM, what you can see is that we validated that we have an increase of the expression of the gene when boosted with DHT. We also looked at whether the effect is mediated by the receptor to androgen receptor. You see that the increase we find, we lose it here when we have the androgen receptor. And this question was asked to us by reviewers, and we did our study on boys with neuronal progenitors, and the reviewers asked us if we could find that in XX or XY, boys and girls, and each time we have this increase of the expression in boys and here in a girl. So this protein, there were other interesting proteins, but this one is a protein that is implied in the axon guidance that enables the neuron to grow and orients it. So we have variants in this gene that were associated previously with ASDs in the introns and promoters of the gene. And in the mouse models, there is a reduction of learning for both sexes and reduction of sociability, but only for males. And in man, there is deletion of the gene with the persons with ASD, and the mutations of loss of function that are implied in the recessive forms of NDD. But here, there's no major difference between boys and girls. No. So what we found in the mouse model, but maybe we didn't look at the proper phenotypes in boys, but there is no obvious difference. And just to complete on this gene, what we did, looking at the variation of the genes, so the receptor is relocates on the DHT. Here you have the NRCAM. We identified several positions on which the androgen receptor will attach to, and the small stars here are associated with the ASD. We would have loved to have something that is exactly located on the ASD associated. And we also found an enrichment. We looked at the genes that are regulated by these beyond the NRCAM. Do we have genes that are mutated, variants that are associated with ASD or NDD? We found a significant enrichment among the target genes associated with ASD. And we also found a significant enrichment in the genes known to be implied in the monogenic forms of ASD or NDD. And we also found an enrichment in the genes identified that are targeted by the androgen and dysregulated in the brains with ASD, with a special module related to inflammation, which was described a few years ago. To conclude, I wanted to show you the work that we did and that was done by an English team in the development of brain organoids, that is a bunch of cells that will differentiate in vitro. And Madeleine Lancaster, the differentiation of pluripotent cells into organoids, and she's tested these cells with testosterones. So this is the visualization of a brain organoid that you can obtain in vitro, and you see here these bubbles that with neurons that will differentiate all around. So each of these bubbles will present what is happening in the cortex, and you can monitor the number of neurons that are produced in different conditions. So what they wanted to see is whether the and they also looked at the effect on the effect of regulation of the regulation of genes. So you have a zoom here, and what you see is that we have different types of neuronal progenitors, which are the glial, the intermediate progenitor, radial glia, basal radial glia. And here on the surface, you have the neurons that are differentiated in the green area. And you can look at these different areas and see if we have a difference in the thickness that shows if there is more neurons or more progenitors. So this is what they did to compare the organoids made of boys XX, boys XY, and XX for girls. And you see that there is no significant difference between any of the regions, boys and girls, at the basal state. And then what they did is that they treated with DHT these cells, and they used a marker called Ki67 and to monitor these cells that proliferate, and TBR2. And what they showed is that at 35 days of differentiation of 52 days, DHT enables the increase of the number of progenitors that multiply during the proliferation phase. So this is consistent with what we showed in our model. And they went further. It's a bit nicer. And they took the organoid and they did some single-cell RNA sequencing. In the control groups in red, organoids treated with green. In the different categories of progenitors that you find when treating with DHT. So they found more or less what we found, that is an increase of the proliferation of progenitors when treating with DHT. So they also found the expression of genes. One of the slides has disappeared of the presentation, but they find an overexpression of the mTOR pathway. And we also had a gene that is implied in this pathway. So maybe something should be investigated further on the activation of this pathway by the androgens. The excessive boys with NDD is not explained by the simple genetic forms identified so far. So we still have to find something, maybe by looking at the non-obvious things in the genome, because we still have things to find on the X chromosome. And maybe there are also complex forms that imply variants of the X chromosome. Some studies are exploring this. So far, the results are not extraordinary. And the sex bias is not explained yet. And the hormone hypothesis is that the androgens lead to an overproliferation of the neuron progenitors. And it was shown by our team and Madeleine Lancaster's team in 3D. In an increase of the mTOR pathway that could explain this overproliferation. And Eric Courchaine, who is working a lot on the development of the brain, and he showed that there would be an overproliferation at an early stage of development. So maybe there is an overlap with people with ASD. So the androgens regulate the expression of mutated genes, namely this NRCAM gene, which we need to investigate further. Even though for the moment the sex-dependent effect demonstrated in mice has not been demonstrated in men. The androgens that the girls do not show the same clinical manifestations as boys, but when we put testosterone, this TCC gene will cause that. So far, there is no answer from genetics to this question of why the excess of boys with NDD. But we see here that we can have an overlap between what is dysregulated with or regulated by androgens with people with and without ASD. The models that were used, our models are not perfect, and in the organoids, the brain organoids, we're missing a lot of cells. What we use is not vascularized. We are missing microglia, so we need more complex models. So, and I wanted to thank my team. The work on androgen, Angélique Cartier really worked on this, and Jean-Louis Mandel, who was the one who encouraged me to work on this hormone hypothesis, and all my colleagues in genetics, clinics, and thank you for your attention.